kindly. Hi, this is Miss Litton, and this is my wonderful and excellent Honors Bio Unit 3 review. Say hi. Hi. I was supposed to be at lunch, but my bad, I forgot. And here we are at 7th period. So I'm sorry if you missed it. Here is the recording. Okay, so Unit 3 was energy and enzymes, right? And it was also cellular respiration and photosynthesis. So let's do some rundown of this unit. What are the laws of thermodynamics? Let's start off with that. And the first one is energy cannot be created or destroyed. It can just change forms. And then the second law says every time it changes forms, you lose some as heat. Perfect. How do they relate to entropy and energy? Well, let's think about what entropy is. Everything is going to greater and greater what? Disorder. And you and I and all living things have to apply energy to avoid entropy, right? Things will break down, so that's why we need energy in order to avoid that. ATP is the energy currency of every cell, right? Adenosine triphosphate. And that's the money that you spend in the cell to protect yourself against entropy. And so there are some strategies that are used in order to either make the fuel source to make ATP or make the ATP directly. And we'll talk about that. Okay, different kinds of reactions. Exergonic and endergonic. Which one sounds like energy is leaving? Exergonic, exactly. So in an exergonic reaction, you're going to lose energy. In an endergonic reaction, you're going to have to put energy in. Where you get that energy from, it could be from ATP. You could be like some catabolic reaction that you have to build, and so it requires ATP. Or you could get it from a different reaction. You could get it from an exergonic reaction. You remember when we talked about reactions that are coupled together? So that would be another way. Catabolic, anabolic. Which one is building? Anabolic is building, and catabolic is breaking down. Okay? So what prevents an endergonic reaction from spontaneously occurring? What prevents an endergonic reaction from spontaneously occurring?
Activation.
No, but an endergonic reaction requires what? Energy by definition. So you just don't have energy around, right? So you need to have specific input of energy to make an endergonic reaction occurring. Now, what prevents an exergonic reaction from just spontaneously happening for the most part? Now you want to tell me what?
Activation.
Yeah, activation energy. Because we looked at an endergonic reaction requires the input of energy. An exergonic reaction, you lose your free, remember delta G, your measurement of free energy, you lose it. So what prevents it from just doing this spontaneously? And that's because even your exergonic reactions have a hurdle right here called the energy of activation that prevents them from spontaneously, let's say, decomposing. Now, to lower that hurdle, you could just add heat, because heat makes all molecules move faster, increases their kinetic energy, and so it would make the reaction more likely to occur. But if you want, that would make all reactions that were exergonic occur. If we want to have a specific reaction occur, we need to use a what? Starts with an E. Enzymes. And enzymes will lower the energy of activation. So that's how enzymes work, is they lower the energy of activation and make a reaction more likely to occur. Now, how can you regulate them? Because enzymes are proteins. They have specific temperatures they work at, specific pH they need to work at. But what was another way we could regulate enzymes? Do you remember? There were inhibitors. Remember the two ones we talked about? Anybody? Competitive and non-competitive inhibitors. You're like, I don't know, kinda remember. Competitive inhibitors, to understand those, we need to know what an active site is. Yeah. An active site is that area on the enzyme that the substrate binds. So if my enzyme name was Handis joinus, okay, joinase, excuse me, Handis joinus, then I would use my hands to join people together. My active site is not my ears. You do things to my ears, nothing's going to happen, right? But my hands are the active site. So if something else gets in my hand, a competitive inhibitor, that would prevent me from being able to do my job. That's hand disjointed, okay? If you have a secondary site somewhere else, and that you bind to, like you stab me in the ear, okay? And now my hands are busy pulling that knife out of my ear. Really good analogy. I'm sorry. I'm super tired. Okay? Now you've affected my active site only because I'm responding to something else, right? It's changing the shape of that protein when some other molecule binds to it. Did I teach you the word allosteric site? Okay, that's secondary site. Good. Competitive, non-competitive inhibitors. We also talked about, yes?
Non-competitive is when it has the allosteric site?
Yes, the secondary site. Competitive if it binds exactly the same place. Like in this case, my hands are the thing that binds to the substrate. And if you put something in my hands, right, that's competitive. You're competing for my hands. Then there was something called feedback inhibition. Do you remember that part? Where you could have enzyme 1 makes A go to B. Enzyme 2 makes B go to C. Enzyme 3 makes C go to D, and so on and so on and so on. And I'm making this product H. If I get enough H, it goes back and interacts with enzyme A to stop the whole sequence from occurring. Have fun. Is it another dry run? Yeah, dress rehearsal. So it would then prevent the whole series. That's called feedback inhibition. So I have enough of my product, so stop it at the first enzyme, not the second or the third. Stop it at the very beginning. Okay, number 4, what is the difference between being oxidized and reduced, and how does it happen? We learned a lot about that in the next chapters, right? So, you want to remember that electrons equal what? Energy. And whoever has the electrons has the energy. If you're reduced, you're high in energy but reduced in charge. What would lower your charge? Electrons, because electrons have what charge? Negative. So you're reduced in charge but you're high in energy. If you hook up with hydrogen, hydrogen tends to give up its electron, and so you will be reduced. Conversely, if you break up with hydrogen, you will become what? Oxidized. Okay? Oxygen is very electronegative, tends to pull on electrons, so if you hook up with oxygen, you will be oxidized. If you break up with oxygen, you will be reduced. And what we see in both cellular respiration and photosynthesis is a series of reductions, right, in our na-na-na-nas. It doesn't matter if we're in a mitochondria or chloroplast. A series of reductions, and along the way of that series of reductions and along an electron transport chain, you're able to capture the energy temporarily in order to move hydrogen ions from one side of a membrane to another. To the outermost chamber for mitochondria, to the innermost chamber for photosynthesis. It's the concentration of hydrogens that creates a chemiosmotic gradient. The hydrogens want back. And how do they go back? Through their doorway. And their doorway is called ATP synthase complex, and it generates ATP. So we see that pattern again and again. Okay, questions on 6. All right, do you feel strong? Okay, let's move on to the 7-8 combo. Okay, 7 and 8, lay them straight. You want to know each of the equations, the steps in each of those equations. There was, for instance, in cellular respiration, there was aerobic and anaerobic, right? In photosynthesis, there was cyclic and non-cyclic. The best one between cyclic and non-cyclic is non-cyclic. Electrons come in via water. They go out via reduced NADP. The best one, most efficient in cellular respiration, aerobic or anaerobic? Aerobic. Good. And then we'll go through those. Am I still recording? Yes, I want to continue. Okay, there. Okay, we'll go through each of the steps. We'll review it in song. But photorespiration, that was specifically about the dark reaction, the Calvin cycle, the light independent reaction. And the first thing we had in there, preloaded into that cycle, was the 3C5. That is RUBP. RUBP is supposed to hook up with our 3C1, our 3 carbon dioxides. But if you are low in CO2, because you've closed your doors of your leaves, your stomata, okay, then O2 builds up and you lose CO2. And then RUBP will be a gardening tool, not a rake, okay? And hook up with oxygen instead of CO2, and we call that photorespiration. And there were some adaptations to avoid that. One is fix a bunch of CO2 at night, so you have it available during the daytime when you need it. And those were called CAM plants. Another one is isolate your dark reactions, right, in the bundle sheath cells. And PEP in that one as well would bind with only CO2, not oxygen, and escort it in. And did I give you another one? I think that was it, right? Those are your ways to avoid it. Okay, perfect. So now let's do song, okay? This is what we're going to do. We're going to talk about glycolysis. And glycolysis then goes into transition prep. Glycolysis in the cytoplasm, everything else in the mitochondria, right? Then an electron transport chain, which generates a bunch of ATP, right? And also the end product of that is also water. Oh, sorry, Krebs cycle and electron transport chain, thank you. Krebs cycle in the matrix, electron transport chain along the membrane. We're going to talk about photosynthesis. We'll do the light reaction and then the dark reaction, and then we'll go back and do anaerobic respiration. Okay, you ready? The whole thing is called life. And one we're going to start out with is cellular respiration. What's the first step? Glycolysis. Who does it? All cells. Where do they do it? In the cytoplasm. What do you start out with? Glucose. How many carbons? 6. What are we going to do? Break it in half. How many steps? 10. How many enzymes? 10. What are these called? 2 pyruvic acids. And you reduce some NAD and make some ATP. 2. Oh, we have plenty of oxygen. What should we do? Aerobic respiration. What's the next step called? Transition prep. Here we go. And you reduce some NAD and 2 acetyl-CoA, put 1 away. Why are we putting 1 away? Because we're going to put 1 through and then the other one. What's the next step called? Another name for it? And when we do the bit, what are we doing? Reducing NAD and pulling off the CO2. Here we go. 2 plus 4 is 6. Do the bit, get 5. Do the bit, get 4. Make an ATP. Reduce an FAD. Reduce an NAD. And 2 plus 4 is 6. Do the bit, get 5. Do the bit, get 4. Make an ATP. Reduce an FAD. Reduce an NAD. And stop. Gather all your reduced NADs and FADs and take them to the top of the ETC. Pause. Why? Some are. Yeah, and they come down a little lower. Here we go. Reductase, cytochrome oxidase, and you make some ATP. A lot of it, or a lot of it. Who catches electrons at the bottom? Water. Oxygen. Who catches electrons at the bottom? Oxygen, forming water. Water. What are we going to do? Break it in half. What do we get? Oxygen and electrons. Sweep. Pause. What are we sweeping? Hydrogen. Where do those come from? Water. Where do these electrons come from? Water. We are in photosystem 2. Here comes the sun, traveling in waves. Particles of light are called photons. And it gets the electrons excited. PQ. And you make some ATP. Here comes the sun, traveling in waves. Particles of light are called photons. And it gets the electrons excited. Who catches it? NADP, forming reduced NADP. Then you take some ATP and some reduced NADP to the dark reaction, or the Calvin cycle, or the light-independent reaction. 3C5 plus 3C1 mix. 3C6 break down to 6C3. Use some ATP, use some reduced NADP on your 6C3. Take 1 away, PJI, G, 3P, and you're left with 5C3. Use some ATP to build
3C5 plus 3C1 makes 3C6. Break down to 6C3. Use some ATP, use some reduced NADP on your 6C3. Take one away, glucose. And you're left with 5C3. Use some ATP to build 3C5. Stop.
Pick up your glucose. How many carbons?
6.
What are you going to do?
Break it in half.
Steps?
10.
Enzymes?
10.
Called?
2-pyruvic acid.
And you?
Reduce some NADP and make some ATP. 2.
Oh no, we don't have any oxygen. What are we going to do?
Fermentation.
How many choices do we have?
2.
You and I make lactic acid.
Makes our muscles sore.
Plants and yeast make
Alcohol. Either way, you oxidize your NADs.
Yay! Good job. Thus endeth the lesson. Study. Have a piece of toast. Be ready. You got this. I love you. All right.